Bismillah ar-Rahman ar-Rahim. Now we start the second lecture in the physiology for Physiotherapy Faculty for non-Arabic speaker students. And our topic today is transport across cell membrane. You remember last time we mentioned that the cell is an thing that's surrounded by a membrane, a cell membrane that things can pass in and out of it. Now
Yes, sir.
we know. Okay, back to the lecture. The cell membrane are made of phospholipids. Phospholipids are the main structural component of the cell membrane. In its name, the description of it, it says that it is phospholipid. Lipid is it's lipid and it has phosphorus in it. That makes it having a characteristic which is very important for being in the cell membrane. It's being both hydrophilic,
Lipid.
can deal with water, and hydrophobic, can
Okay.
deal with water. The part directed inside inwardly in the cell membrane, this is the hydrophobic part. The part outside is the hydrophilic part.
Okay.
The phospholipid goes with other components like cholesterol and glycolipids. There is also some proteins in the cell membrane. As we mentioned earlier, the proteins in the cell membrane has main six functions: transport, intercellular joining, enzymatic activity, cell-to-cell recognition, signal transduction, and attachment to the extracellular matrix. So now is the main topic of the lecture today, how the structures move in and out of the cell membrane. There is three main families or main categories of the transportation. That's the passive transport, active transport, and both exocytosis and endocytosis. To summarize it, the passive transport, there is no energy needed. No energy needed at all. But for the active transport, that's an active process. We need ATP, we need energy. And for the exocytosis and endocytosis, we will talk about it later on. So, passive transport, that's the net movement of molecules and ions across the membrane from higher to lower concentration down the concentration gradient, and it doesn't require metabolic energy. It doesn't require energy. When I ask in the MCQ about transportation that doesn't require metabolic energy, you cannot choose active transport because it requires metabolic energy. So active transport is the net movement across the membrane that occurs against the concentration gradient, and yes, it requires ATP and a carrier protein. Simple diffusion, there is substance that can move with simple diffusion that doesn't require energy, and there are substances that can't move with diffusion and requires energy.
Okay.
Those who can move are the non-polar molecules like fatty acids, vitamins A, D, E, and K, steroids, oxygen, carbon dioxide, and water. Those who can't move the polar molecules like polysaccharides, amino acid, glucose, nucleic acids, and ions like sodium, potassium, calcium, and hydrogen.
Okay.
As you see over here, there are simple diffusion through protein channels. The substance move down concentration gradients through the protein channels without requirement of metabolic energy, without requirement of ATP. So, simple diffusion through protein channels, the forces affecting the direction and rate of the diffusion are mainly two forces. The two forces are concentration gradient and electric gradient. How is that? We will act like that's the cell membrane. That's outside, that's inside. If the substance over here, the yellow substance, we will call this substance the mahamiho. This is substance is called mahamiho, and it is much outside. So the rate of diffusion will be high because the gradient is very different between outside and inside. So
Inside.
we'll go is that way, that way, that way, that way, that way.
Okay.
The same goes with the if, again, that's the cell membrane. outside is 2 positive electrical gradient and inside is 2 negative electrical gradient, so the substance will move that way down the electrical gradient with high rate. Also, the factors that affect the rate are 5 factors: temperature, distance of diffusion, size of molecule, surface area of membrane, membrane permeability. And what is that? The membrane permeability is the ability or the concentration or the percent of the membrane that are able to permeate substance. So if the substance if the membrane has many protein channels, those are highly permeable membrane, as many substance pass through it. But if the membrane is less permeable, the rate will be less for the diffusion. As for
Okay.
the surface area of the membrane, if we are having a surface area like this and another one is like this, that's very high surface area to make diffusion that can occur here or 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 here. But for this one, only here, only here, only here. So increase of surface area increases the rate of the diffusion. Also, the size of the molecule. Would you think that the larger the size, the higher the rate, or the smaller the size, the higher the rate?
The higher the rates. The say it again. Sorry, doctor.
If the
The higher
molecule
the
is
price.
too large, it will be faster, or if it is too small.
It is too large.
Actually, يعني, if the molecule is like, for example, like
Okay.
this, and
Okay.
another molecule is like this, which one will be faster or higher in rate, do you think?
The big one.
No. The small
No.
one is light, is small, it can pass fastly. But this one,
Okay.
too heavy, too
Okay.
slow. It takes much time to go inside.
Okay.
So, also the distance of diffusion. If the membrane is too big, there is much distance to be covered for diffusion. And also, when there is higher temperature, the molecules gains energy from the heat and the diffusion rate increases.
Oh.
That's a common question. What is the facilitated diffusion? It is a diffusion, so no need for metabolic energy. The no need for metabolic energy, so it is not active transport. It is facilitated diffusion. It is a diffusion down concentration gradient that doesn't require ATP, but yet it requires a carrier protein that allows substance to go from inside to outside or from inside to outside. And the example for such organic large molecules that needs facilitated diffusion are glucose and amino acid.
Okay.
We talked about diffusion, we talked about facilitated diffusion. Now some talking about osmosis. Osmosis is the net diffusion of water from region of high water concentration to the region of low water concentration through membrane which is permeable to water but not to the solute. So if the water moves down concentration gradient, down concentration gradient, down concentration gradient without need for metabolic energy, without need for ATP, that's osmosis, and that's something that happens naturally down concentration gradient on all cells in all membranes. So again, you think that يعني, the factors that affect the rate of diffusion will also affect the rate of osmosis, and yes, you are right. So if the temperature is higher, do you think the osmosis rate will be higher or lower?
It's going to be lower.
Sorry.
It's going to be higher.
Again, if the temperature is high, the osmosis rate will be
Lower. Yes. Definitely.
so. Let me ask about the diffusion. If the temperature is high, the diffusion rate will be
Lower. No, higher, higher, higher. Sorry.
higher, doctor. So if the temperature is higher, that diffusion rate will be higher. And the
Yes.
same goes with osmosis. If the temperature is high, the osmosis rate also will be higher.
Yeah. Okay.
There is something called osmotic pressure. Osmotic pressure is when yeah, it can be the alternative of concentration gradient in the case of diffusion. Osmotic pressure is the force that would have to be exerted to prevent osmosis, and the greater the solute concentration, the greater the osmotic pressure. Pure water has zero osmotic pressure because it has no solute, it has no solute concentration. What we call saline or 360 gram per liter glucose solution has twice the osmotic pressure of the 180 gram per liter glucose solution. The higher the solute, the more the solute, the higher the osmotic pressure. Now we talk about osmolarity, and the osmolarity is the number of osmoles per liter of water, and that's mean, and you have to be in much focus during the exam because we have osmolarity and osmolality. The osmolarity is the number of osmoles per liter of solution. The osmolality is the number of osmoles per kg of solution. What does that mean? It means that there is a power of osmosis in each solution or weight of fluid. And the normal osmolarity of body fluids are 290 milliosmol per liter. That number is for memorizing. You should go into the exam memorizing such number.
Okay.
So again, we mentioned osmolarity and osmotic pressure. We will talk about isotonic solutions. The isotonic solution is solutions that have the same concentration of solutes as the extracellular fluid. Hypertonic is the higher the concentration that the inside. Hypotonic is the having lower concentration than the inside. So again, I will explain with such drawing. If we have a test tube and a test tube and a test tube, the three test tube has a solution, and the three test tube has an RBC inside them or a cell inside them. If the solution is isotonic, so the osmolarity in the solution is equal to the osmolarity of the cell. If the solution is hypertonic, so the concentration in the solution is much higher than inside the cell. If it is hypotonic, hypotonicity means the solution is low in concentration than lower than the cell. Is it clear?
It's clear.
Okay. The movement of some substance through the cell membrane, we talked about fast diffusion in all its terms, simple diffusion, facilitated diffusion, and osmosis. Now we will talk about active transport. Again, the most important differentiation between active transport and diffusion is the need of energy in active transport. It is an active process that needs metabolic energies, that needs ATP, so it is against concentration gradient. It requires specific carrier protein and utilization of energy. Types of active transport are two: primary active transport and secondary active transport. What is the difference between primary and secondary? The secondary, that it doesn't only needs what we mentioned as carrier protein and energy, it also needs substance that to be transported, co-transported or anti-transported, counter-transported. We will talk in that a minute after now. The primary active transport is one of the most important structures to allow the normal state of body, and we have a channel called sodium potassium pump in the cell membrane. That's the primary example of the primary active transport. The sodium potassium pump uses ATP energy to pump 3 sodium outside and 2 potassium inside. That's an example of primary active transport channel, and it uses energy, it uses carrier protein, and it pumps those two ions against their concentration gradient. The secondary active transport, for transportation of two solutes through the same transporter protein, it can go counter transport or co-transport. The most common example for that is the SGLT, sodium glucose potassium sodium glucose transporter channel, co-transporter channel in the kidney or in the intestine, and those channels are very important for conditions like diabetes mellitus. We have the drugs to treat diabetes mellitus by يعني inhibiting those channels to inhibit the reabsorption of glucose and allow glucose to be reduced in blood. So again, you don't need to know all those information. You just need to know that the secondary active transport is a transport occurring at the cell membrane that requires energy, requires protein, and requires two other solute to be transported, co-transported or counter-transported during the process. The co-transport or the symport is an example of, as I mentioned, sodium with glucose co-transport at the border of the intestinal or sodium amino acid transport at the intestine. The counter-transport are the sodium calcium counter-transport or sodium hydrogen counter-transport. You just need to memorize the example and the type of the transport. Again, to summarize the active transport, we have the primary and secondary. Secondary can be counter or co-transportation. Finally, we have the endocytosis and exocytosis. Endocytosis and the exocytosis are transportation occurring at the cell membrane, and that transportation is a transportation that transports large molecules that cannot pass through any channel in the cell membrane. So we create a small vesicle that can engulf from outside or exocytosis from inside. So the exocytosis is removing outside, endocytosis is engulfing into the inside. Endocytosis can be in two types, phagocytosis and pinocytosis. Phagocytosis for solid substance like bacteria, like large substance or debris. Pinocytosis is for fluid, and we call it also cell drinking, which is example of transportation of fat or protein inside intestinal cell. As for exocytosis, we also remove substance, vesicles that contain calcium or any other thing outside the cell membrane. Thank you. I hope it was helpful.
Very, very good.
Do you have any question?
No, no, sir.
Alhamdulillah. Inshallah, I hope to meet you soon to finish the remaining lectures. Inshallah. May Allah bless you and see you soon. Bismillah.
Thank